Good evening, everyone. Okay. So we'll wait for a few more minutes if anybody else joins, and then we'll initiate the session. So, yeah, let's begin the session for today. Okay. So, in today's session, we are going to discuss the genome editing methodology using the zinc finger or nucleases. Okay. So, zinc finger nucleases. Okay. Basically, we are going to talk about this. Before initiating this topic, let us go to the basics. So this zinc finger nuclease is a protein. Okay. So protein, we know it is the basic unit is the amino acid. So the amino acid join each other by the bond that they form, and that forms a linear chain of amino acid. Okay. The amide bond, peptide bond. Okay. So they are joined by peptide bond and made a linear chain of the amino acid. So this is called the primary structure, primary structure of the protein. Okay. So a certain set of the proteins, they are joined together, make one functional protein. Okay. So now this protein, in order to achieve stability, undergoes folding. Okay. That means more compaction of this linear structure. Okay. So for more stability and accommodation inside the cell, it undergoes folding. So that folding also, it has several levels. So once these primary structure or this linear chain of amino acid, they join each other, they can make a helix. Okay. They'll make a helix, or um, or they also make interact each other and make a sheet kind of structure. Okay. It could be a helix, it could be a sheet. It is the helix referred as alpha helix and the sheets are referred as beta sheets. Okay, so these compaction occurs via the hydrogen bond formation. Okay, so as we know the amino acid structure, so it has a carbonyl. So this makes this peptide bond, right? When the two amino acids join. So in the alpha helix, what happens? Uh, there are several amino acids. So the first amino acid with the fourth amino acid, the carbonyl oxygen and the fourth amino acids, this amine hydrogen. Okay, they makes a bond referred as hydrogen bond. Okay, so they join each other. These amino acids are joined each other via the hydrogen bonding, and that makes this primary structure into a helical form. Okay, so that makes a helix that referred as alpha helix. Similarly, if the structure of the amino acid is arranged in a beta, it's in a sheet form, plated sheet form, then it is a beta sheet. Okay, there also the similar hydrogen bonding takes place. Okay, so in beta sheet we have um, so in alpha helix, in the same primary linear chain only, the intramolecular hydrogen bonds are happening. However, in beta sheets, generally two amino acid chains are involved. Okay, so one one chain of amino acid, another chain of amino uh, acid, and they are joined together, making a beta plated sheet. So one amino terminal this side, carboxy terminal this side, another amino acid, the amino terminal this side and carboxy terminal this side. So, and however here, if we'll see, both they are in the orientation the same side. So these are called the parallel beta sheets, and these are called anti-parallel beta sheets. Okay, so these are the secondary structure of any amino acid. Okay, secondary structure. Then after the secondary structure, so after the secondary structure, here comes the super secondary structure. Okay, so secondary structure is nothing but these two different types of secondary structure. For example, helix and sheets are joined together and makes a kind of motif. Okay, so motif is a part of super secondary structure. Okay, so it could be like an helix, then comes a beta sheet, then again one helix. Okay, so it is helix, beta sheet, and helix. Okay, so this is a fixed kind of super secondary structure which has some kind of role in the protein's entire structure. Okay, so the protein has several type of secondary structure and then undergo a from secondary to a tertiary structure. Okay, tertiary structure, which is a more compact form. Okay, where other than hydrogen bonds, these secondary structure are associated together and makes a proper tertiary structure of a protein. In this tertiary structure, there are certain structures which are kind of conserved. So they are called motifs. Okay, so these are the motifs that has some particular function or some particular role or uh, function or a role involved or designated. For example, there is a super secondary structure or a motif which is helix turn helix turn helix motif. Okay, so this is one. So where you'll have one alpha helix Then you have certain amino acid which will make a turn, which is not making any bonds or something. Then again another helix will come. So this is called helix, turn, and helix. So this is a motif, which is a part of the tertiary structure of the protein. Okay. So if this helix turn helix structure is present in your protein structure, that means that your protein has some affinity towards DNA. If these kinds of motifs are present, they are called as DNA binding motifs. DNA binding motif. That means if this structure or this motif is present in your protein has ability to bind to DNA. Okay. So this is a DNA binding motif, helix turn helix. Okay. You can there is a difference between the motif and the domain of the protein. Okay. So with the motif, you can predict a protein's role, what could be the role or where it could be involved, but you cannot associate motif with its function. Okay, if this helix turn helix motif is present in your protein, that means your protein can bind to DNA. But upon binding, what role it will perform, that is not known in case, or that cannot be claimed in case of motif. Yeah. However, in case of domain, so domain is an independent functional uh, structure of a protein. Okay. So one protein has multiple domains towards N-terminal, a certain entire tertiary structure of a protein, a 3D structure that makes a domain. And domain can specify a function of the protein. For example, there are ATPase domain. If that particular structure is present, or that kind of, then it is referred as a domain. And if it is a ATPase domain, that means you can tell that your protein can hydrolyze ATP. So that is the function of your protein. So that domain can specify the function, but the motif cannot specify the function. Motif can only predict about its role, what could be the probable role. Okay. And two domains are independent of each other. If your protein has multiple domains, if you mutate one of the domain, the other domain or the function of other domains will be unhampered. Okay. So that is the difference between the motif and a domain. Okay. So this is a DNA binding motif, which is generally present in the proteins which are interacting with the DNA. Okay. So now let's talk about this protein, one of the DNA binding protein where these kind of helix turn helix motifs could be present. So, okay. So there is a protein which is FOC1. Okay. FOC1 is the endonuclease. Endonuclease. Okay. So this is a FOC is derived from Flavobacterium oceanocortex. Okay. From there it has been isolated, and it is an endonuclease, which is an unusual type 2 endonuclease. Okay. So and generally type 2 nucleases are the nucleases which recognize a particular DNA sequence and makes a cut there. However, this FOC1 endonuclease is an unusual type 2 endonuclease. That means it recognizes a specific sequence but makes a cut certain bases away from the recognition site. Okay. So I'll draw. So it has a recognition site which is then you have several bases. Okay. Okay. So this is the recognition site. This from 5 prime to 3 prime. GGATG is the recognition site for this endonuclease. Okay. It recognizes this nuclease, but the cut will be made in the 5 prime strand from 9 base pair away from the recognition site. Okay, here 9 base pair away. Here somewhere it will make a cut. And in the 3 prime strand from the recognition site, 13 base pairs away from the in the 3 prime strand. Okay, so this is unusual type 2 restriction endonuclease fork 1, which has a particular specific recognition sequence, but the cleavage is non-specific. That means it make a staggered cut away from the restriction site. Okay, so this is the fork 1 endonuclease. This is because in this particular protein structure, it has two regions. Okay. This is N terminal, this is C terminal. So the N terminal is the recognition, is for recognition site. Okay. And the C terminal is the cleavage site or the catalytic site of this particular enzyme. And they are linked together with a sequence which is called linker sequence. Okay. So this is the structure of this particular protein. Okay. So when this particular enzyme is present, the recognition site recognizes the sequence, which is GGATG from 5 prime to 3 prime. The recognition site will sit here and recognize, and the other protein, which is the catalytic protein, will sit away from the recognition site, the cleavage or the catalytic site. Okay, so this protein is a dimeric protein. It is a dimeric protein. Okay. So once one recognition site or one of the monomer gets bound to the DNA, another monomer of this protein comes and recognizes the sequence on the other strand. Okay, and then it happens. So this recognition site comes, and this is the cleavage. First one, second one. Then another monomer comes and recognize the sequence, and with the help of cleavage site, with the help of the alpha helices, alpha 4 and alpha 5 helices, which are the parallel helices, these two makes the active fork 1 enzyme. And this is the active form. Only one monomer attached to the DNA is the inactive catalytic form of the enzyme. When both the monomers dimerize to each other on the DNA strand, then it makes an active fork 1 enzyme, and then it makes a cut on the DNA. Okay. So this is the story of this particular protein, fork 1 endonuclease. So scientists have discovered that this particular cleavage site, as because these are two different domains, so their function is detachable from each other. So this recognition site can only help to recognize, but the cleavage site, it makes a non-specific cut. So if you can remove this portion and replaces it with some other protein that will be specific to your DNA sequence, then that fusion will work, and you can make a specific cut at your desired location, and this cleavage site will make a cut there. Okay, so that because of this modular nature of this particular restriction enzyme, scientists now introduced other recognition site proteins so that they can recognize and bind to the DNA, and the cleavage, non-specific cleavage site will make a cut there, so on. Okay. So then this particular protein comes, which is some another kind of DNA binding protein, which is having a zinc in its active site. Okay. So a study on the protein transcription factor 3A from Xenopus oocytes. Okay. So from this particular Xenopus oocytes 5S rRNA, this particular protein was identified as a transcription factor. So here a repetitive zinc binding domains are present, which further referred as zinc finger. Zinc finger. So here, a study of the transcription of targeted RNA sequence revealed that the binding strength of a small transcription factor 3A, TF3A, was due to the presence of zinc coordinating finger-like structures. Okay. So amino acid sequence of TF3A revealed 9 tandem sequences of 30 amino acids, including 2 invariant pairs of cysteine and histidine residues. Okay, so those are called zinc finger. Okay, so these zinc finger kind of motifs are also DNA binding motifs. So if in your protein structure this particular motif is present, that means it has ability to bind to the zinc DNA. Let's see further. So 1940, the biological role of zinc in proteins was established, and it was shown to participate in carbonic anhydrase catalytic activity. So it was found that zinc can play a unique role not only for CA but also for other enzymes catalytic activity. Okay. So the structural role of this particular zinc was proposed for the Xenopus laevius transcription factor 3A. Just now I showed you the paper. Okay. So the Xenopus laevius transcription factor A structure consists of repeating 30 amino acid residue folded around zinc ion to form a small, highly independent domain called a finger. So why it is called a finger? Because you can see, so it is a beta beta alpha structure. Okay, 2 strands of beta sheets. Okay, you can see 1, 2, and 1 alpha helix, and there the total 30 residues are included. Okay, 28 to 30 residues are included, and it makes a finger like of structure. Okay, that is why it is referred as zinc finger. Okay, so a beta hairpin which comprise of anti-parallel beta sheets consisting of 2 beta strands. Okay, consisting of 2 beta strands followed by an alpha helix together form a left-handed beta beta alpha structure. Okay, so this zinc finger structure is stabilized by the coordination of zinc atom with the 2 conserved cysteine residues and one end of the beta sheet. You can see here in the beta sheet, one end of the beta sheet, 2 cysteine residues. You can see here, here in this particular picture, 2 cysteine. So these cysteines are present in the beta sheet, and with 2 conserved histidine residues at the alpha helix C terminus. Here in the alpha helix, these are the histidine residues. Okay. So other amino acids which are present in this 30 amino acid fold are a consensus sequence which says that they are more or less similar but not exact identical. So they can vary, but these cysteine and histidine are the invariant amino acids and conserved through all the this C2H2 domain of type of zinc finger proteins. Okay, so this is the classification that has been done. So it is a C2H2, 2 cysteine and 2 histidine. We saw. Okay, cysteine, histidine, bound together with this zinc atom, which in short we refer as C2H2 of like finger. Okay, C2H2 like finger. Other we have gag nickel, treble clef nickel, zinc rubber, which is a classical zinc rubber, rubidoxine family. Okay, so there are certain classifications, but all have this finger-like structure where the residues may differ but have the similar function binding to the DNA. Okay, so each of this finger, okay, so if in a protein that in this particular paper it was reported now, repetitive zinc binding domains, repetitive uh, finger like structure. So in one protein, like suppose 1, 2, and 3 finger like structure joined together with the help. So as I mentioned, 30 amino acid finger like structure. So out of 30, usually 25 out of those 30 are involved in making this finger like structure, and remaining 5 are usually work as a linker to join it with other finger like motif. Okay, 5 out of 30. This is a generalized way. So in a protein, certain finger-like structures are present, followed by each other, which recognize a 3 base pair specific sequences. Okay. For example, this particular finger-like structure where those 4 cysteine and histidine residue are conserved in all these 3, but the others varies. So because of those variable residues, the recognition differs. So the finger one can recognize this sequence G, A, T, 3 nucleotides, so 3 bases. And other finger recognizes A, C, T because of the variable amino acids present in its finger like motif. Another recognizes T, C, G. But this recognition is from 3 prime to 5 prime, unlike the 5 prime to 3 prime direction. Right? So, a zinc finger protein consisting of 3 linked zinc finger modules. It binds to its target DNA site with amino acid of the recognition with the recognition of alpha helices into the N to C terminal direction, contacting consecutive nucleotides and DNA running in 3 prime to 5 prime strand. So, as we saw in the structure, so this particular alpha helix, it recognizes the DNA. So, this particular alpha helix recognizes the DNA, the double helix DNA, and that also because of the this alpha helix can insert itself into the major groove of the DNA, and there the binding and the interaction will takes place between the residues, active residues in the alpha helices and in the major groove, those bases which are present. Okay, yeah, like this has mentioned. So this is the alpha helix. As we mentioned, it is a beta beta alpha structure. So the beta is not represented here. Okay. So this is the beta beta alpha. So in the alpha strand, so the amino acid -1. So if this is the 3 prime to 5 prime, 3 prime to 5 prime. So as it was mentioned here, GAT. So suppose it is a GAT. Okay. So this in the -1 from the alpha helix, that one recognizes the third amino acid G. The from the alpha helix +3, third amino acid recognizes the second base, and from the alpha helix the sixth amino acid recognizes the third base towards the 5 prime. Okay, so that is how the recognition will take place, the interaction will take place. And this second from the alpha helix, which recognizes the another base that is present in the 3 prime strand. So this is the specific kind of recognition pattern. Okay, so each of the 3 C2H2 domains bind to 3 or 4 DNA nucleotides via amino acid at the same alpha helical positions. Arginine at position -1, as well as amino acid residue as position 2, 3, and 6. Biochemical and structural studies of the C2H2 domains confirm the key role of the amino acid at these positions for the specific binding to the DNA. According to the canonical model, the amino acid at the position 6, 3, and -1, 6, 3, and -1 are responsible for the recognition of the first, second, and third nucleotides at the 5' end, respectively. And the amino acid at the position 2 recognizes the fourth nucleotide on the complementary strand. Okay, so this is how the recognition happens. Okay, so then the modification was done. Okay, so by changing these amino acids, a high degree of selectivity can be achieved towards a given 3 base pair DNA sequence. Okay, so exploiting this recognition mechanism, protein modules containing multiple zinc finger motifs, each one recognizing a specific 3 base pair DNA sequence, have been engineered, which bind to specific DNA sequences. Okay, so that means, as we have shown here, this is the sequence of a DNA. What you can do, you can design several zinc finger protein, modulate with its the amino acids which are helping in recognition, okay, and then design your zinc finger protein according to the DNA base you wanted to interact, okay, and link several such zinc finger and make your recognition from non-specific to a specific recognition. Okay, I hope you are able to understand what I'm trying to say here. So fusing the recognition module with a sequence independent endonuclease was the first successful strategy to introduce breaks at specific sites of the genomic DNA. So that, what does it mean? In the restriction endonuclease, what we have learned that uh, that it recognizes a sequence, the restriction endonuclease. Generally, they recognize 4 to 6 to 8 base pair sequences. Okay, and after recognition it will make a cut there, but as our genomic content or the DNA sequence has a nature of repeatedness, so this particular um, 4 base pair sequence which is being recognized by this restriction nucleus can be repeated several more times. Okay, so that will create several non-specific breaks throughout our genome. So to be more specific, a longer stretch of DNA recognition enzyme is needed so that we can make a cut at the desired site in our genome. For example, uh, okay, for example, there is a gene. Okay, in the genomic locus, there is a gene. Gene name is CNSP1. Okay, now you want to do manipulation in this particular gene. You can't just select a 4 base pair, 4 nucleotide recognition enzyme, because those 4 nucleotides will occur several base pair away in this region as well or further away. That will make several double strand breaks non-specific. That's why you have to be specific for that. Ask me any doubt. If you are not able to understand, stop me anywhere. Okay, and try to interact, please. Okay, so suppose this is the gene. Now it has a specific sequence. Okay, so what we can do, we can design a protein which can recognize a long stretch of the sequence, which is present in this particular position in the genome of the organism. Okay, if it is a very long sequence, its probability of being present at some other genomic locus will be very less or null. Okay, because you can cross check that. So the longer the sequence, the more specific would be the recognition. Okay. So you can make your enzyme. So as we are utilizing that zinc finger proteins, okay. So zinc finger protein recognizes only three bases. So you have to make a chain of such zinc finger proteins so that your recognition protein becomes more specific. Okay. So you make a protein like this. Okay. So this particular zinc finger motif recognizes these three bases. Another protein recognizes these. Next series recognize this. Another one will recognize this. Now you have recognized the location where you want to make a cut. Okay. Now this recognition protein, you can fuse it with other protein, which is a cleavage protein. Okay. Which is a cleavage protein. Now after the recognition, this will create a cut here. Okay. So this cleavage protein should be non-specific. Now after the recognition, now this gene is cut in two pieces. And that is how the specific recognition is happening. Okay. So this is recognizing the specific sequence, the recognition protein, and you have fused it with a cleavage protein, which is an endonuclease, which will create a cut once the recognition takes place. Okay. So that will make a cut. Now your protein is fragmented. You can exploit this break to make a mutation, insert a mutated copy at this broken junction, or another gain of function mutant copy. Everything can be done here. So that is what. Fusing the recognition module with the sequence independent endonuclease. So we read about the sequence independent nuclease from the fork one. Okay. In the fork one, we have two regions. Okay. The N-terminal region, which was for recognition and which recognizes the sequence, and this C-terminal region, which is a non-specific endonuclease, which will just make a cut. Okay. So you can replace the recognition sequence with this a long stretch of zinc finger like protein harboring several repeated zinc finger structures in a protein, and that fusion protein will now work for specific making a specific cut at the genomic locus in your desired organs. Okay. So from basic research to translational research. So this zinc finger nucleases. So now after the fusion, it is referred as zinc finger nuclease, because zinc finger protein, it just have the ability to bind to the DNA, zinc finger protein. And the fork one, which also recognizes its recognition sequence, is also a very small sequence. So for fork one to be more specific, we can fuse it with the zinc finger protein, and that together makes a zinc finger nuclease. Okay. So now this zinc finger nuclease resulted from the basic research on the zinc finger proteins and the fork one restriction enzyme. So a zinc finger protein consisting of three linked zinc finger modules. Generally, it is a three zinc finger modules. The first discovered one. Three linked zinc finger modules bind to the cognate DNA site and carries out activities like transcription. Okay. So as read in the paper, the transcription factor 3A, that it works as a transcription factor because it binds to the DNA. And the fork one has a bipartite structure with a separable DNA binding domain and a non-specific cleavage domain. So zinc finger nucleases are the synthetic chimeric proteins composed of specific zinc finger DNA binding domain linked to a non-specific fork 1 DNA endonuclease cleavage domain. So these are the zinc finger nucleases, which you can exploit for your particular desired genome editing. Okay. So let's address the questions for today. Try to get involved and answer the questions. So the classical zinc ribbon belongs to which fold group? In the beginning, I showed you the table where the classification was done. There was a classical zinc ribbon also. Yes. Option? Option D, right? Zinc ribbon. Yeah, the classical zinc ribbon belongs to the zinc ribbon group. Okay, so the zinc fingers were first identified in a study of transcription in the African clawed frog. What was the name of the frog? Yes, Xenopus laevis. So how many repeated amino acid sequence does a typical C2H2 zinc finger motif contains? So as I already mentioned it, yes, it is 28 to 30. Generally 30. In this particular one, 28 to 30. The correct option. So which of the following is the rate limiting step in the homology directed technology for gene modification? Okay, let me first tell you this. Simple. In the last class, we discussed about homologous recombination, right? Where you exploit the homologous recombination machinery to make a desired uh, modification at your genomic locus. So once the double strand break is made in your particular gene of interest, okay? So this is the double strand break made here. This is your gene starting site. This is ending site. So you can exploit this break via the two method. One is the homologous recombination mediated machining. So what you will do in your gene of interest, you suppose you want to make a mutation in this particular gene. So what you can do where the you have make a break in the functional domain in provide a homologous template copy. Okay, a homologous means the sequence similar to this, sequence similar to here, and in between that you introduce the desired modification or the mutation that you desire to do in this particular gene. Okay, and once the break is created and homologous template if it is provided, the cell will shift to make or use HR as a more efficient machinery because it is very less error prone, it is highly specific, and it will incorporate the correct copy. So when the homologous template is provided, cell will use the homologous recombination machinery and use this template to repair this double strand break via the homologous recombination pathway. Via getting the homologous region here, the HR will be active here, and now in your particular gene of interest, this extra mutant, this extra mutant is now incorporated here. Okay, so this is a knock-in approach. Okay, this is a knock-in approach. You make a cut, give a template which is homologous with a little variation or mutation, desired mutation and modification you want to do, and then cell will make a double crossover, use the HR machinery, incorporate that thing to repair it a broken end, and that is now, so this is the wild type, and now after the break in HR, it has become a mutated gene. Okay, it is a mutated gene. So now your gene of interest is mutated. You can study what is the consequences of this mutation in order to survival or in any pathway. You can study the phenotypes. Okay, so this is how you can exploit the double strand break. Other than that, if not HR, if there is no template strand, what will happen? Non-homologous end joining, where there is no template is there. So what generally, so HR is a very error prone. Okay, because exonucleases are there are chances something will be chopped off, some X portion will be chopped off, and your DNA or the gene, when ligated back, it has lost some of the DNA sequences that were present in between this. Also, uh, there are chances that some extra non-specific thing will get integrated, and that will also be, so either deletion or insertion. In the both the cases, the uh, your gene will be affected. So this is non-homologous end joining, just to see the effect of the uh, gene disruption. This is basically you are disrupting. It will lead to deletion, insertion. What will happen, that you can study. Or if you provide a homologous copy, homologous template, that will integrate it, and that will be very specific. Whatever mutation you want to study, deletion you want to study, the domain you can remove and insert. So that will be a specific study. Okay, you can opt for any of these two. But the very crucial step, the rate limiting step, making a cut at the desired location. Okay, so that desired location, we are troubleshooting, and we are overcoming this problem. We can overcome this problem with our zinc finger nucleases. The zinc finger helps in the specific recognition of the uh, DNA site where you want to make a break, and the nucleus or the endonucleus from the fork one will create a break after the recognition. So the very important thing here is the making that particular double strand break, which is specific to your study. Okay, so now if I'll ask you to answer this question, which of the following is the rate limiting step in the homology directed repair technology for the gene modification? Gene targeting, is that the rate limiting step? Generation of non-specific desired genomic DSP, or generation of specific desired genomic double strand break, or none of the above? Which would be the answer? Yeah, generation of a specific desired genomic double strand break. That is crucial and a rate limiting step. Good. Okay. So once these zinc finger nucleases are designed, then their delivery inside the cell, okay, the culture, the mammalian cell, the tumor cells, whichever cell line you are working with, how will you deliver that zinc finger nucleus? One thing, when you when you make, you just basically add the DNA fragments. So that particular DNA construct, you can clone it in a vector. That could be a viral vector. So viral vector can incorporate or when go inside the cell and can replicate on its own, make several copies, and your work will be done. Use a viral vector. There are other non-viral vectors also, which includes these liposomes, cell-penetrating peptides. Other than that, you have the physical methods that includes microinjection, electroporation. Okay. So electroporation is nothing, just a short electric pulse that will open the cell membrane for a short time. That pulse, so that your particular construct can enter in that short duration inside the cell. That is electroporation, giving electropulse. So these all the methods can be used for the successful gene delivery. Okay. Other than that, the direct um, insertion of the protein is also possible, because there is a positive C2H2. The positive, because of the positive charge of the C2H2, directly the zinc finger nucleus protein can also be transferred via the cell membrane. Yeah. So what is ribosomal skipping? Okay. So in this particular modification, So we are using the recognition site from the zinc and making from the zinc finger proteins and the cleavage site of the fork one. Okay, so fork one, you remember we talked, it is a dimeric protein. Okay, it is a dimeric protein. So dimeric proteins also um, base. So zinc finger recognition and then a cleavage site. Okay, similarly, so this needs to be dimerized, right? So on the other strand also, those zinc fingers will be required, right? So, but the sequence will differ. Okay, if I'll write any sequence. Mhm. Okay. So from 3 prime to 5 prime, one zinc finger, finger 1 recognizes GAG, other one recognizes CAT. The third finger will recognize this. This is one stretch of the DNA has been recognized from this particular portion. But the dimerization is needed. And the other dimer will bind to the other strand from 3 prime to 5 prime in this on the opposite complementary strand. But there the sequence will differ, right? So here, as here GAG, CAT and GTC present, here GAT, TAT, GGC, other sequences present. Okay. So for that you need another protein. Okay, another protein that will make another zinc finger nucleus. So this is zinc finger nucleus 2 and this is zinc finger nucleus 1. Two different protein you have to overexpress inside the cell so that both of them go recognize the site and then make a cut. Okay, here it will make a cut. So making or expressing two particular proteins, what could be done? You clone this particular construct, this particular ZFN1 protein in one vector, ZFN2 protein in another vector. Okay, one vector ZFN1, another vector you have cloned ZFN2. Okay, now you have to make both the vectors enter into the cell, enter into the cell. So there are probabilities sometimes because of the limitation of the transformation transfections protocol, if both the uh, this vectors are not able to enter, okay, or one particular have entered, more copies of this one has entered into the cell and less copies of this one is present in the cell, then also there is a problem in making the break. So to ensure uh, the expression of both the nucleases in the cell at the same time at the similar level, what you can do, make them clone in a single vector. Okay, so in one vector itself you clone the zinc finger nucleus 1 and the zinc finger nucleus 2. Both of them you can clone in the similar vector and you can put them under the same promoter. Put them under the same promoter so that its expression will take place in the your organism. Okay, but you don't want the fusion protein to be made because they are placed under the similar promoter, they are placed in the similar or in the sequential manner. What will happen once this particular protein will be made, Along with this, the another protein will also be made. So that is called a fusion protein. Two proteins can fuse with each other. And then fusion you don't want. You want the separate monomers so that they can recognize two separate sequences. By recognition, they make a dimer, and that will be active catalytic enzyme that will create a cut at the desired DNA sequence. So in order to make two different or co-translation of two different proteins at the same time from the same promoter, what is there is a peptide, that is a viral peptide 2A. Okay, it is a viral peptide 2A. That peptide sequence you can put in between the two proteins, two nucleases. Okay, so this viral peptide, what does it do? It does the translational skipping. Okay, so sorry, sorry. No, no, it is not poly A. That A is not poly A. That is as from the discovery they are numbered. So that is the second number of that peptide that is discovered from that virus, and that is the numbering 2A, 2B, maybe that. Okay, from there it has come. It is not the poly A or 2 adenine. Okay, it is a peptide which has the name 2A. Okay, yeah. So yeah. So in this peptide, the RNA will be made. This is one protein, and this is another protein. Okay. So the RNA will be made. So this is the 2A sequence, and when the translation will takes place, the translation will takes place when the ribosome will start making proteins from there. So when so it start making protein from the first one, right? So when this particular peptide comes, it doesn't take the upcoming amino acid to the ribosome. Doesn't allow the ribosome to take the upcoming amino acid, that prevent that next amino acid addition to the ribosome. And it is a self-cleavable peptide. So once this sequence reach, it cleaves the chain here at this position. Okay, that cleaves itself with the leading or the growing amino acid chain. Cleaves itself, and then this remaining RNA is free to undergo the second round of translation, and that will make another protein from here. Okay. So from the same locus, from the same clone, now two particular proteins are ready. One of this ZFN2 and another protein ZFN1. So now two nucleases which has different recognition sequence are co-translated from the same vector with the help of 2A viral peptide. Okay, so this is the method. So that particular practice is called the translation ribosomal skipping. So ribosomal skipping is the mechanism of translation in which specific viral peptide prevent the ribosome from covalently linking a new inserted amino acid and let it continue translation, resulting in co-translational cleavage of polyprotein. So this is the correct option. I hope it is clear. So, which of the following statement about direct delivery of ZFN proteins is true? Yeah, it can penetrate the cell membrane. So the cysteine and histidine has a positive charge on them, right? So due to the presence of the positive charge in this zinc finger domain, they can penetrate the cell membrane. So that will be the correct option. Which of the following statement about zinc gene deletion using zinc finger nuclease is true? Gene deletion can be achieved by single zinc finger nuclease mediated double strand break. Is it correct? Is it correct? No. So what else can be done? Just so as I have mentioned only about the double strand break in between the gene, right? So what you can do, you can design two such zinc finger nucleases, one at this place, one at this place. This will make break here. This one will break here. And now with the two double strand breaks, your gene of interest is completely lost after the break is created. Okay, it is lost. Then it can be joined by the NHEJ pathway. Okay, so this is a knockout of gene. So two such zinc finger nucleases are needed to make the gene deletion. It's clear? These for using this also you can achieve the deletion. Just now in between the gene that I told you to make the break, that can help you to introduce the mutation, or you have to rely on the non-homologous end joining to uh, make a deletion by the help of the exonucleases. Okay, so you can use two zinc finger nucleases to make a double strand break that will lead to the gene deletion. Okay. Next is gene deletion cannot be achieved using zinc finger nucleases. True or not? It can be achieved, right? Two concurrent zinc finger nucleases mediated DSPs can result in a loss of a gene. This is the correct option, right? Two concurrent zinc finger nucleases mediated DSPs can result in the loss of gene. This one. It's clear, right? Shall I move to the next one? Which of the following strategy a pair in with which of the following strategy a pair of zinc finger nuclease can be cloned and co-expressed together in a single vector? Okay, you tell me the answer. Introduction of a 2A sequence. Yes. In the presence of homologous sequences, homology directed repair result in that. Site specific integration and gene editing. Yeah, this is the yeah, this is the correct option. It is a site-specific integration that will lead to the gene editing. Okay. Thank you. Anything you want to ask me? Was everything clear to you? Do you have any doubt? I can't hear you properly. Can you speak a little closer to the mic? Yeah, tell me. Yeah, yeah, yeah. Yes. So I have explained the questions. So basically, I have to make you practice the questions, what kind of questions are there. So related to that, all the things I have told you. Is there anything that I left? Okay. So every live session, we are covering 1 week's portion. So in the week 5 portion, you have the zinc finger mediated genome editing, right? So only that portion I'm talking today. The other parts are talked in the next weeks. So it is a weekly class. I think that is what you were asking, right? Mm-hmm. Mm. No, I have covered from both the lecture 1 and lecture 2. Lecture 3 and 4 are the basic applications, which are very detailed, uh, taking the one particular organism, and that is also similar. I will tell you the similar. The strategy is similar, only the application is shown. For example, in Drosophila and some other mammalian cell lines, taking a specific gene name, right? So that is why I didn't touch that part. But the basics and the basic strategy, how the zinc finger nucleases are work, that I have taken today. Okay. Yeah. Anything else? Okay. There is no question. I'll conclude today's session. Thank you everybody for joining. Okay.